Today's lab will be on aesthetic techniques. My name is Viviana Soto and my lab partner is Kaytlyn Torres. And we are doing a lab in our VTPB 426 microbiology lab. The main objective of this lab was to learn and practice aesthetic inoculation techniques, which means transferring microorganisms from one medium to another without contamination. This is one of the most important skills in a microbiology because it allows us to keep pure cultures pure and ensures that our results are reliable. In this lab, we work with different types of media, including broths, slants, deeps, and plates, to see how bacteria grow in each environment. We also use different tools like loops and needles depending on the type of medium. Overall, this experiment gave us hands-on experience with aesthetic technique and showed us why it is essential for studying and working with microorganisms. For this experiment, we worked with pure cultures of Escherichia coli in broth and Staphylococcus aureus on blood agar plate, as well as a mixed culture broth. The media was used included tryptic soy broth, TSA slants in both 3 mm and 9 mm sizes, and TSA plates. To transfer the bacteria, we used inoculating loops and needles. A burner was also used to help maintain sterile conditions. These materials gave us everything we needed to practice aesthetic inoculation across different types of media. So we used different tools because the lute and the needle helped us spread organisms on the surface of the agar or the broth, while the needles deliver microbes into a deep agar where oxygen conditions can vary. The loop and needle must be sterilized between streaks to help avoid transferring too many cells, ensuring colony separation and preventing cross-contamination. Um, Inoculation differed. Broth required simple transfer with the loop. Slabs were streaked on the surface. Deeps required a stab with the needle, and plates required streaking from isolation. So some precautions was working near the burner flame to reduce airborne contaminants, flaming tube mouths, sterilizing tools before and after use, and keeping the lids closed when not in use. The media included broth to observe turbidity and general glow, the slants to see surface growth, the deeps were to test oxygen tolerance and motility, and the plates for colony isolation and morphology. So for the procedure, we practiced transferring microorganisms into different types of media using aesthetic technique. We used inoculating loop to place the E. coli and the S. aureus into broth tubes and streak the TSA slants. For the TSA deep tubes, we used a needle to stab into the agar and to streak the slant as we pulled it out. On the TSA plates, we streaked for isolation using E. coli, S. aureus, and mixed it with culture broth, which allowed us to separate individual colonies throughout the process. We sterilized the loop and needle between transfers and worked near a burner to help, like I said, sterilize the conditions. So for our results, here are our results for the TSA plates. The first plate showed the mixed culture where we can clearly see two distinct colony types. This demonstrates the importance of streaking for isolation. Without separating the colonies, it would be difficult to study each species on its own. On the second plate with the Staphylococcus aureus, we observed large round yellow colonies. However, the streaking wasn't perfect and there was some overlap between growth areas, so this made it harder to get fully isolated colonies, but we could still see the typical colony morphology of the S. aureus. The third plate showed Escherichia coli, which the colonies were smaller and cream colored. Organic streaking wasn't ideal since some areas showed heavy growth instead of the clear isolated streaks. Even with that, we were still able to see the difference in the appearance between the E. coli and the S. aureus. Overall, these results highlight both the value for proper streaking for isolation and the challenges when technique isn't precise. Even with some streaking errors, the distinct colony types were still visible, which helped us compare pure cultures to the mixed cultures. Moving on to the broth cultures, both E. coli and S. aureus grew well. We could tell because the broth turned cloudy, which is called turbidity. This shows that the bacteria multiplied and spread evenly throughout the liquid medium. On the TSA slant with S. urus, we saw clear growth along the streak line where the loop had been used. The colonies followed the inoculation path, which confirmed that the transfer was successful. The growth pattern also matched that we expect on a slant. It's surface growth that stays mostly where the inoculating tool made contact, unlike the broth, where the bacteria spread throughout the medium. Slants like this are useful because they give us like a solid surface to observe colony characteristics while also being compact and easy to store. For the 3 ml TSA slant with E. coli, we saw growth along the streak line where the loop was drawn. This observation that the transfer was successful, it also showed how the slants provided a solid surface for the bacteria to grow in a controlled pattern. In the 9 mm TSA deep with E. coli, we observed growth both along the stab line and the surface of the slant. The pattern shows that E. coli is capable of going throughout the medium, meaning it can tolerate different oxygen conditions. This deep medium is essential useful for seeing how bacteria behave in the areas of less oxygen compared to the surface. The 9 ml TSA deep with S. urus showed a very similar pattern growth along the stab line and at the slant surface. Like the E. coli, this indicates that S. aureus is a facultative organism that can grow in both aerobic and less oxygenated conditions. Overall, these results not only confirmed that our inoculation technique with the loop and the needle worked as expected, but also gave us insight into the growth capabilities of each organisms in different environments. In conclusion, this lab gave us hands-on practice with aesthetic inoculation and overall techniques worked as expected. Using different tools and media allowed us to see how bacteria grow in different environments from both slants, deeps, and plates. The mixed culture plate was especially important because it showed why streaking for isolation is necessary. Without isolation, the different species would blend together and we wouldn't be able to tell them apart. By streaking correctly, we could separate colonies and identify individual organisms, which is essential in both microbiology labs and clinical settings. However, we did have some errors while streaking, especially on the S. aureus and the E. coli plates, which made it harder to isolate individual colonies. Still, the results showed the differences between the two organisms and reinforced the proper um, technique. Looking ahead, there are a few improvements we could make. We could make more precise streaking would give us clearer isolation of colonies, which makes results easier to interpret. Careful handling during transfers would reduce the chances of contamination and ensure the growth patterns reflect the intended organisms. Finally, using a larger sample size would give us a stronger, more reliable data to compare across groups. This lab emphasized the importance of aesthetic technique in microbiology. Without it, contamination could make results unreliable or even unsafe. Overall, the experiment connected directly to why aesthetic technique and streaking for isolation are fundamentally important in microbiology and clinical work study. Thank you.